Good morning. It's November 1st when I'm making this in the year 2020. So, this is a quite interesting story. We're going to see some stories on it in class, but if you think about it, it's not hard to observe that offspring resemble their parents. Puppy dogs, people, pine trees, offspring resemble their parents in ways that makes it clear that their parents somehow gave them something that made them look like them and even act like them often, at least to a degree, right? But to figure out what that was took a long, long time. Not till the mid 1900s did we humans even figure out that it was this chemical that parents give their kids that causes them to have brown hair or blue eyes or whatever. It's this chemical. That took until the mid 1900s to figure that out. And then once it was figured out that it was this chemical, to figure out how it worked, it took another little bit of a while, but until you figure out what your parents actually gave you, there's no way you can figure out how it works. So we're going to look at a couple of those experiments. They're not in the EOC syllabus, but they are for your information and will be on your tests, at least a little tiny through the rest of the notes. No animals were harmed in the making of that photoshopped photo. I don't know why I threw that in there. No, you can stop and look at those. So here was one of the experiments. I think this was like 1930s, something like that. I'm getting old and forgetful, but 1930s or so. This guy, Frederick Griffith, was not even working on this question of what is the genetic stuff. He was working on pneumonia. He was a pathologist trying to figure out something about how pneumonia worked. So this is a kind of pneumonia that was caused by bacteria cells. These bacteria cells come in several different varieties. We call them strains or varieties, right? Here they call them strains. It's kind of like breeds of dog, right? They're all the same, but they have some characteristic differences. Here you have the S kind that have this surrounding called a capsule, and they can cause pneumonia. So when he injected them into some laboratory mice, it killed the mice. They had pneumonia. Okay, not surprising. And then their cousins, the R type, we'll call them here, their cousins that don't have a capsule are harmless in terms of causing pneumonia. Injecting mice with them, no pneumonia. It seemed obvious that this capsule had something to do with the ability to cause pneumonia, because otherwise these bacteria are the same. Just the presence of a capsule is the difference. So now he took some of the bad guys, right? We're going to call these the bad guys now, the pneumonia causers. He took some of the bad guys, heated them up until they were killed, injected them into mice, and no surprise, right? If they're dead, they can't cause pneumonia, and so the mice didn't get pneumonia. But then the key part of his experiment was when he mixed the dead bad guys, the heated up bad guys that he had already shown didn't cause pneumonia, and the guys that were alive but didn't have a capsule, and he already had showed that they didn't cause pneumonia. When he injected them into mice, the mice got pneumonia and died. And when he took some blood out of them, he found something that he didn't put in them. He found living pneumonia causing with a capsule kind of bacteria. And he hypothesized that somehow these dead guys had passed the ability to make a capsule to those live guys, and then the live guys made the capsule. They passed that capsule making ability onto their kids, which he took out of the blood of these mice. And so this was the first experiment to show that some stuff can get passed on from one to another. He had an idea that this stuff was that chemical called DNA. The chemical DNA had been discovered in the 1860s. Very big year for biology, the 1860s. The discovery, 1859, I think, maybe specifically, by a German guy, Friedrich Miescher, and he actually named it deoxyribonucleic acid. Nucleic acid, because that's where he found it in the nucleus of cells. Had no idea what it did, though. In fact, had no idea at that time what the nucleus really was kind of all about and what it did. So that's the first experiment. Then in 1944, this is University of Chicago, I believe, big time research college. These guys did some experiments building on Griffith's, which is how science also works, and they announced that they figured out that what Griffith had called the transforming factor, he named this process transformation, which we still do, was this chemical DNA. But biologists were still skeptical that one experiment would have solved this mystery about all of life. So they had done all their experiments with bacteria cells, and so rightfully so, you know, that you shouldn't accept that DNA is the hereditary stuff for all of life if you've only tested bacteria cells. So here was the set of experiments that clinched it. So this brings into play some different kind of subjects, and here's where it might get a little confusing to you with some vocab again. So this is a bacteria cell, right? This is a bacteria cell. This is not the pneumonia-causing kind, so this really doesn't have that much to do with the Griffith experiment. And here are some viruses. These very spaceship-looking things are viruses that infect bacteria cells. Now you see DNA labeled here, but this was not known at the time in 1952 when Mr. Hershey and Miss Chase did this experiment at Cold Spring Harbor in New York. This is on Long Island in New York, still a very big famous research institute. And what was not known at the time was, what did viruses put into cells, like this bacteria cell, that caused these cells to make more viruses? Because it had been figured out, also in Cold Spring Harbor, how viruses work. They convince cells, so to speak, to make more viruses. They can't reproduce by themselves. They get a cell, which we call a host cell, to reproduce them. Somehow they give the instructions to reproduce them to these host cells. And it's those instructions which were being looked for. What are the instructions? The two biggest candidates among biological molecules for the hereditary instruction stuff was DNA, or nucleic acids in general, and protein. The question was, which one was it? Well, life is so complex that for a good while, proteins were the logical favorite because proteins were known to be much more variety and much more complex than DNA. DNA seemed like a pretty simple, ordinary kind of molecule. So we'll get back to that later. But so that's an actual micrograph like you see there. Definitely electron micrograph, right? And this would also be a transmission electron microscope that took that picture. Colorized, right, because it wouldn't have been in color, so it's been colorized. So here's the experiment that we're going to watch in class and talk about just a little bit. One of the things that's confusing about this whole story is that these viruses are called bacteriophages. Oops, Phages. Phages for short. That's what you see them called here. It's kind of phage. There you go. Right. That's the little spaceship-looking viruses, which would be way smaller than bacteria cells relative to them than they have shown here, but for whatever. Okay. Bacteriophage. Remember we had phage, phagocytosis. This word literally means bacteria eater, which is also not a great name because they don't eat bacteria cells. They use them to reproduce. But anyway, bacteriophages are not bacteria cells. They infect bacteria cells. Once you keep all that vocab straight, here's the story, okay? They took some of these viruses that they had made up to have a radioactive protein as part of their outer coat. These viruses have an outer coat, this was all known, an outer coat and these like landing gear, literally, what these are, that's all protein. So this is all protein. And then inside, they have DNA. So these viruses were great subjects for this experiment because they were only made of the two kind of molecules that were being considered as the hereditary stuff. And it was known that they somehow put instructions to make themselves into these bacteria cells, host cells, that they landed on. So was it their DNA that they put in them, or was it their protein, or maybe it was both, right? So they had a batch of viruses that had the protein coat radioactive. Then they had another batch of viruses that had the DNA radioactive. And in separate little compartments, they let the radioactive protein viruses infect bacteria cells, and then in another separate little set of test tubes or petri dishes or whatever, they let the radioactive DNA viruses infect bacteria cells. Then after they let them have enough time to send into the bacteria cells whatever they were sending in, they put them in a blender. And they did that because they wanted to separate the bacteria cells and whatever virus stuff they had inside from the virus stuff that stayed outside. And then they separated all that and they said, okay, which batch of bacteria cells has radioactivity in it? The ones that were infected by viruses that we made the protein radioactive, or the ones that we infected with viruses that had the DNA radioactive? And indeed it was the ones with the radioactive DNA that they found inside the bacteria cells. This told them that this is the stuff that bacteria cells can take and make new viruses with. This is therefore the hereditary stuff. This is what a virus passes on to its kids, so to speak, with the help of these host cells. This is what a parent passes on to their kids that gives them the instructions to make them like their parents. So this is the one that did it. That was 1952. So then, in just a few places in the world, because there's not a lot of scientists who are trained and qualified in this kind of biology, right? They started to try to figure out, ooh, DNA is the hereditary stuff. This is like the biggest thing in biology, right? Because biology is the study of life. This turns out to be the chemical that gives life all its characteristics. This was like as big as can possibly be in the world of biology. So now all eyes, so to speak, turned on DNA to try to figure out how it worked. And that started with, what's its structure? To figure out the function of something, from molecules to whatever, you have to figure out its structure first.